Hey there. Every single living thing, you, me, your dog, that plant in the corner, is built from this incredibly detailed instruction manual called DNA. It's a code, and it dictates pretty much everything about us. But what happens when there's a typo in that code? Well, today that's exactly what we're going to unlock as we dive into the fascinating and super complex world of genetic disorders. So what's the game plan? First up, we're going to figure out what these genetic typos actually are. Then we'll put on our detective hats and see how scientists track these traits through family trees. After that, we'll zoom way in on disorders that are caused by just a single misplaced letter in our DNA, before zooming back out to see what happens when entire chapters of the code go wrong. And finally, we'll tie it all together with a nice, clean summary. Sound good? Let's go. So really, our whole conversation today revolves around this one big question. If you think of your DNA as the ultimate biological blueprint, what happens if a single letter is wrong? Or a whole page gets copied incorrectly? What are the real-world consequences? That's what we're here to unpack. All right, first things first. Let's talk about our genetic typos. We need to get to the very heart of the matter and really understand what a mutation is. Okay, so what is a mutation? At its core, it's just a change in our DNA sequence. Simple as that. Now you'll see two words here, genotype and phenotype. Think of the genotype as the specific genetic code written in the instruction manual. A mutation changes that code. The phenotype is what you can actually see, the physical traits that result from that code. This one process is the engine behind everything, from genetic diversity to disease. Now, these changes can happen on totally different scales. You can have a point mutation, which is like a single letter typo in a giant book. And we'll see exactly that with sickle cell anemia. Then there are frame shift mutations, where letters get added or deleted, which just scrambles the entire sentence that comes after it. And then you have the big one, chromosomal aberrations. This is like entire chapters of the book being duplicated or ripped out. Okay, so we know these typos exist, but how do we actually find them in the human story? Well, that brings us to our next section. We're going to explore how scientists track traits through generations using this really cool tool called pedigree analysis. You know, Gregor Mendel, the father of genetics, could crossbreed thousands of pea plants to see how traits were passed down. Obviously, we can't do that with people, right? So scientists use the next best thing, pedigree analysis. This is basically genetic detective work. You build a detailed family tree to watch how a specific trait or disorder moves through generations. It lets us see the patterns of inheritance play out in the real world. And this right here? This is our decoder ring for reading those genetic family trees. It's actually pretty straightforward. A square is a male, a circle is a female. If a shape is filled in, that person has the trait we're studying. A horizontal line connecting a square and a circle means they're a couple, and the lines dropping down from there lead to the kids. That double line means the couple are relatives. These simple symbols are like the universal language for human genetics. And here's how we put those symbols to work. It's awesome. So look at chart A on the left. That's a dominant trait. See how the disorder shows up in every single generation? If a parent has it, there's a good chance a child will get it. Now compare that to chart B on the right, a recessive trait. Notice how the parents in that second generation are totally fine, but they have a child who is affected? That's the telltale sign. It means they were both carriers, and the trait skipped a generation. Now that we know how to track these things, let's dive into our first big category of disorders. This is where we look at single gene disorders, when just one tiny typo can change everything. We call these Mendelian disorders, and they're caused by a mutation in just one single gene. And the absolute classic case study for this is sickle cell anemia. Check out this chain reaction, it's incredible. It all kicks off with one tiny point mutation. The DNA code GAG changes to GUG. That one little switch causes the wrong amino acid, valine, to get plugged into the hemoglobin protein instead of glutamic acid. This one swap is enough to change the entire protein. And when oxygen levels are low, it forces the red blood cells to twist into that rigid sickle shape. A single letter change with life-altering consequences. And here you can see the cause and effect plain as day. On the top left, that's the code for the normal protein, which leads to those nice, round, healthy red blood cells you see. But on the right, that single base change from GAG to GUG creates the faulty protein, and the micrograph shows you the result in stunning detail, those weirdly long, sickle-shaped cells that cause all the problems. Now, just to give a little more context, let's quickly compare sickle cell to another common condition, color blindness. The really big difference here is where the faulty gene lives. Sickle cell is autosome-linked. It's on one of our regular numbered chromosomes, number 11 to be exact. But color blindness is sex-linked. The gene is on the X chromosome. And that explains why it's so much more common in males. See, guys only have one X chromosome, so if they get a faulty one, that's it. There's no backup copy to mask the trait like there is in females. All right, let's scale up. We're moving on from single-letter typos to huge copying errors. It's time to talk about chromosomal scale changes. What happens when entire pages of that genetic blueprint are either missing or added? So, the mechanism behind a lot of these big changes is something called a nucleotide. It sounds complicated, but it just means the gain or loss of one or more chromosomes. Usually it happens when something goes a little haywire during cell division and the chromosomes don't separate properly. The result? Cells end up with the wrong number of chromosomes. Down syndrome is probably the most well-known example of aneuploidy. The cause is an extra copy of chromosome 21, which is why it's also called trisomy 21. And you can see it right there in this picture. This is a karyotype, which is basically a map of a person's chromosomes. If you look at the spot for chromosome 21, you can count them 1, 2, 3 copies instead of the usual 2. That extra genetic material is what leads to the condition's characteristics.
And this number really hammers the point home. A typical human cell has 46 chromosomes, 23 pairs. But for someone with a trisomy like Down syndrome, the grand total is 47. Just one extra chromosome, but it changes, well, everything.
Now, this whole aneuploidy thing doesn't just happen with the regular chromosomes, it can also affect the sex chromosomes, X and Y. And here we see diagrams for two of those conditions. On the left, we have Klinefelter syndrome, which affects males, and on the right, Turner syndrome, which affects females. Let's break down the genetics here. Someone with Klinefelter syndrome has an extra X chromosome. Their genotype is XXY. This results in a male who is usually sterile and might show some feminine development, like gynecomastia. Now, Turner syndrome is the opposite. It's caused by the absence of a sex chromosome. The genotype is just X0. These individuals are sterile females with underdeveloped ovaries. It's a perfect illustration of how both the gain and the loss of a chromosome can have massive effects. Wow, we've covered a ton of ground, from single letters of DNA all the way up to entire chromosomes. So let's bring it all home with our final section, a summary of these disorders and our key takeaways. And this table just lays it all out perfectly. Think of this as your ultimate cheat sheet. You can see the two main categories we talked about. You've got the Mendelian disorders, caused by single gene mutations, like sickle cell and color blindness. And on the other side, you have the chromosomal disorders, caused by gaining or losing whole chromosomes, like Downs, Klinefelter's, and Turner syndrome. It's all about the scale of the error. You know, understanding our own genetic code is one of the greatest scientific adventures of all time. But the more we learn, the more responsibility we have. We're getting to a point where we're not just reading the blueprint of life, but we're learning how to rewrite it. And that leaves us with a pretty deep question to think about. What do we do with that power? Thanks so much for watching.